Invading pathogens or other toxic agents can trigger the formation of inflammasomes by activating sensor proteins such as those in the NLR family. Activated sensor proteins in turn induce the aggregation of a protein called ASC to form large intracellular specks that recruit and activate caspase-1, leading to a rapid pro-inflammatory form of cell death known as pyroptosis. Inflammasome assembly has been visualized in vitro, but Maria Leptin and colleagues wanted to observe this process in a live animal, specifically in zebrafish.
We discovered a huge family of presumed innate pathogen sensors, and we wanted to study them and look at inflammation in a live organism. And it became clear that we didn't have good readouts. And so Paola said, well, let's look at one of the conserved molecules. So we start somewhere where we know where we are rather than starting with lots of unknowns, and made tagged versions of ASC so that she could observe them in vivo. First by a transgene, and then it became clear that we end up with overexpression. So it wasn't physiological. So eventually we decided it was necessary to have a knock-in.
Lead author Paola Curie used CRISPR-Cas9 to generate zebrafish expressing GFP-tagged ASC from the endogenous ASC promoter.
And where we see it is basically in all the epithelia that are in contact with the outside world. So the outer skin of the fish, the gut epithelium, and the gills. But it's also in microglia in the brain. So, these are the guys that scavenge the brain for dying cells. So they're sensitive to danger signals. And also in a subset of myeloid cells.
Curie et al. Saw that ASC specks, a proxy for inflammasome activation, formed spontaneously in epidermal cells during zebrafish development. And the researchers were able to induce widespread speck formation using toxic concentrations of copper sulfate, or by overexpressing either ASC itself or one of the NLR proteins thought to act upstream. Correlative light and electron microscopy revealed that, just like specks assembled in vitro, in vivo ASC specks are composed of highly cross-linked ASC filaments, forming compact globular structures approximately 700 nanometers in diameter. As barriers to the outside world, epithelia such as the epidermis are often the first port of entry for pathogens or other toxic agents, but the role of the inflammasome in these tissues is poorly understood. Curie et al. Saw that keratinocytes die within minutes of ASC speck formation, showing all the hallmarks of pyroptosis. Specks can form in either the nucleus or cytoplasm. Just one speck forms per cell, though, and it's unclear whether nuclear specks are capable of inducing cell death.
So we targeted the ASC to the nucleus. Paola added a nuclear localization sequence, and that induces specks and it kills. What you see when a speck is formed, it soaks up all the ASC that's in the cell instantaneously. So within a few minutes, the cell is really completely depleted of diffuse ASC, and it's all in the speck. So if you see it forming in the nucleus, you can then see that the speck in the nucleus first soaks up the nuclear distributed speck. And then whether it even soaks up the cytoplasmic speck, you can have it both ways.
Specks therefore induce rapid keratinocyte death, regardless of whether they form in the nucleus or cytoplasm. Dying cells in the outer layer of the epidermis can be extruded from the epithelium, but in the basal layer, dying cells lyse and fragment, releasing ASC specks into the extracellular space.
And then we very often see macrophages coming to gobble up the speck. The macrophage can walk around and gobble up a few more, and then either the speck sits around for a long time or it gets degraded. What we've never observed is the speck soaking up additional ASC in the macrophage and killing the macrophage. That's been demonstrated in vitro, but we don't see that.
ASC is composed of two protein-protein interaction domains, a pyrin domain and a card domain, joined by a flexible linker. Both domains can self-associate, and so when Curry et al. Overexpressed the individual domains in keratinocytes lacking endogenous ASC, they formed small filaments or aggregates, but they were unable to assemble into normal compact specks. In mammals, the ASC card domain also binds to the card domain of caspase-1, thereby recruiting and activating this protease to initiate pyroptosis. Curry et al. Confirmed that caspa, a zebrafish homologue of caspase-1, is required for speck-induced pyroptosis, but caspa contains a pyrin domain rather than a card domain, suggesting that the mechanism of caspase recruitment and activation is slightly different in zebrafish cells. Curry explains more.
So if you only have pyrin, you trigger like a bad aggregate kind of thing, but since it's still able to recruit the caspase, even if they're not compact, they will lead to cell death, but with a slower mechanics. If you express only card, then we think that because you're not able to recruit the caspase anymore, then you will no longer have cell death, but the aggregates will still form.
ASC's pyrin domain is therefore necessary and sufficient to induce cell death, but the card domain is required to form a proper compact speck and trigger pyroptosis rapidly and efficiently.
So I think in the paper we generated a lot of tools that can be exploited to look at things in vivo that have never been seen before. And so the things that we are trying to do, we're trying to investigate how the protein behaves in vivo in the context of particular infections. But since ASC has also been linked, for example, to cancer and in some because it propagates inflammation, you can also think of ways in which these tools can be used to study these processes live by using zebrafish.
There are also many outstanding questions about the biochemistry of ASC, such as why it only forms one speck per cell and how the specks soak up the soluble pool of ASC so quickly. For now, though, you can learn more about the dynamics of in vivo ASC spec formation in the paper by Curry et al., published in the September 4th, 2017 issue of the Journal of Cell Biology.